Our next speaker is Ed Boyden. He's assistant professor at MIT Media Lab, where he's a Beness Career Development Professor. He also has joint appointments in the Department of Biological Engineering, the Department of Brain and Cognitive Science, and is the leader of the Synthetic Neurobiology group there. And it's a great pleasure to welcome Ed. And boom, he's ready to go. Thanks.
All right, thanks so much. So our group is chiefly a neurotechnology group. We work on strategies for trying to control and observe what happens in neural circuits in real time, usually at the millisecond time scale or faster. And one of the reasons that we are interested in control is this idea that at every level of abstraction of the brain, whether we're talking about regions and the connections between them, all the way to the electronic microscopic scale, we're seeing things that could go awry. We're seeing things where we don't understand what each of the functions are of each of these elements. And we think of this like the brain like a computer. What we really like to do is be able to observe what's going on at each of these different computational subunits, and also to be able to perturb them and program them in order to test what's going on. And so controlling offers two advantages. One, of course, is that you can, as David already showed, you can try to substitute information into a circuit, or you can try to delete it. And in either case, you can try to sculpt what's going on and test your knowledge of what that information really is doing. The second thing, which must be all too obvious as well, of course, is that if you can control things, you might be able to fix them. And pharmacological approaches, of course, have been one of the great success stories in the 20th century for treating brain disorders, and of course, deep brain stimulation as well. But if we have ever more precise methods for control, then there might be new therapeutic avenues that are opened up. So one question that we've been pursuing over the last several years is just how precise and how systematic can we get with our ability to control neural functions. In order to address this question, we've been turning to nature to look for tools that allow us to speak the language of the brain, but from a remote distance. And it turns out that there are lots of photosynthetic critters out there that convert light into electrical energy. And of course, electricity is sort of the common currency of brain computation. And so much as how GFP and many other molecules from the wild have proven valuable in biology, we've started to try and do this kind of mining as well. And one of the open questions is, of course, how and what directions can you go in terms of this kind of mining of natural resources for new tools for biology. So the story really began about half a decade ago, when in collaboration with Carl Dijsselroth and Gerard Nagel, I showed that you could take a molecule from a green algae, Halorhodopsin 2, which David has already mentioned, and put it into neurons and use it to control neural activity. Now this molecule, which looks something like this little crystal structure there, contains a vitamin A derivative, and therefore can operate and be light activated and absorb light without needing any kind of extra chemical that has to be added. And so one of the usefulness is that it has this functionality where here's a little channel in a membrane here, schematized in yellow. When you shine light on it, it'll open up a channel pore and let positive charge in, which is exactly what would happen in the brain when it fires an action potential. So we were able to create viruses that use this molecule as its payload and to put into neurons, and a lot of pieces of luck aligned here. It expressed well on the surface of these membranes, although most algae and neurons are quite different. And also it was able to mediate currents that were sufficient to mediate high precision driving of spike trains. And since then, a wide number of groups are using optical fibers put into the brain. It's used in a wide variety of species. It's used in transgenic mice. And so on and so forth, that can be used to sort of tease out the sufficiency of a given cell type or a given pattern of activity to drive downstream circuitry in order to perform some kind of behavior or to go awry. Now, we've also started looking for molecules that can mediate neural silencing. And silencing is important because you can test the necessity of something. If you delete it, you might be able to see what it's important for. And if you wiped out all the information in it, then that can be a good way of checking what was required in the context of a circuit. So there's a couple different strategies for how to do this. One is you can take negative charge on the outside of a cell, and if you can shine light and translocate charge in, then that would allow you to shut down neurons. And in late 90s, a paper came out that proved very inspirational, where they showed that electric current as a function of chloride for various classes of molecules called Halorhodopsins sometimes would cross over into the mammalian regime. Now, what do I mean by that? These molecules are from critters, archaea, that live in very high salt concentrations. And so as you can see here, the peak electrical activity happens at very high chloride levels, 5 molar chloride, which is much higher than what's in your brain right now. Most of us have chloride levels probably down around here. And you can see that these molecules don't function very well at low chloride levels. But one of these molecules in this paper was found to be different from the rest and had a peak that, for whatever bizarre evolutionary fluke, happened in the mammalian chloride concentration regime. And so a few years ago, we tried this out, and indeed were able to get quieting currents. You can see a neuron spiking away. If we shine light on it, when it expresses this Halorhodopsin, it will delete those spikes. Now, while these are able to mediate fast transients, the currents are somewhat non-impressive. And there were other issues as well. We found that if you shine light on these molecules for an extended period of time, like tens of seconds, not so long from a behavioral standpoint, they would go into a long-lasting inactive state that would take tens of minutes to recover. And we weren't the first to discover this, but we found this in every Halorhodopsin we've looked at now in many different species. You could use a little pulse of blue light to reset it back to its initial state. And I won't go into the photocycle physics why this is so, but that complicates the optics a lot. So we went through and screened a whole bunch of different mutations in order to try and see if we could accelerate the recovery, and had some trouble doing that. So we thought, well, let's go back to the drawing board here and look once again at genomic diversity. And in a paper that we published earlier this year, we started scanning chloride pumps, opsins from funguses, opsins from bacteria and archaea, and we found some new classes that had really unique, interesting properties. One of these molecules that we nicknamed ARCH was able to mediate order of magnitude improvements in quieting currents, both at low light powers and at high light powers. It's about 15 to 10-fold, depending on the power, enhanced over the original Halorhodopsins. And so what this means is that you can actually have digital silencing. The vast majority of the cells that we were recording here in the wake-behaving mouse are 99.5% shut down or more, as shown in the population data here. There's a bit of a long tail here, which are probably the non-virally infected cells that are just coming along for the ride as we quiet neural network activity. Now, we characterized the electrophysiology of this, and to our surprise, this is actually pumping positive charges out of the cell. So here we have our little membrane again and the receptor, and we shine light on it, the green light in this case, the positive charges are pumped out. And the positive charges are protons. So we started trying to figure out how this worked. Protons are usually thought of as quite rare in neurobiology. pH 7 is in the micromolar scale or less. And so one of the questions is how does it work and what is it doing? We're also trained to fear protons in neuroscience. So we wanted to check to see if it was really bad. So we tried silencing for minutes and didn't see changes in action potentials after versus before the silencing period. We measured the pH changes with some help from John Lin and Roger Chen's group on advice on what to pick. And there were pH changes, but they were very small, 0.1 to 0.15, which is actually smaller than the pH changes that they had found in the Halorhodopsins, which turned out to also be proton pumps and proton channels, and similar in magnitude to the variations in proton concentration that you see during normal neural activity in the cases where it's been monitored in vivo. One last note on these is that you can really, they have much accelerated kinetics. So we're able to do repeated silencing, hundreds and hundreds of trials without any changes in the long-term efficacy, which is great for behavioral experiments. So we've gotten great behavioral experiments in a number of circuits now. But importantly, this ability to resolve different molecular classes is really powerful because they have different properties. One of these properties that came out in the study was that molecules from different kingdoms have different spectral properties. So the Halorhodopsins from archaea were red light quietable, but not as much affected by blue light. And fungal opsins, almost as a rule, were blue light quietable, but not so much affected with red light. Now we're continuing these screens along many different axes. One of these is that we're starting to drill down into the families of molecules to look for more and more powerful variants. Here we have some clues as to where to look for. Let's explore locally in genomic space, where previously in our published work we've been scanning quite globally. So one of these is we've been drilling down to a specific genus, the Halorubrum genus, which had this molecule ARCH. And we've been scanning around for its close relatives all over the world. These are from organisms that appear in China and South America and other places. And what we found was that we could actually find some that were 3 to 5-fold more light sensitive. So this one we've nicknamed ARCH-T. It's from the species TP009. And in this molecule, we could actually get very good silencing in the non-human primate, which has a large brain, and you need to have better light sensitivity. So to summarize the population data here, in a 1 millimeter diameter ball, which is large neuronally speaking, we were able to get a median of 100.0% silencing out of all the neurons that showed any degree of quieting. And the worst case was around 80%. So we're very optimistic about these high light sensitivity silencers being useful for this kind of silencing and also repeated silencing. You can see here 50 trials in a row, and we're not seeing any change in efficacy over time, thanks to the ultra-fast time kinetics here. Here's another example. One of the things we like to do is be able to control neurons at this neuronal scale. We're turning entire neurons on and off. But there's so many different ion channels and receptors, and you heard in Alan's talk sort of a survey of some of the ones that they've been mapping. It would be nice if we could simulate those as well. Now, if you can only turn things on and off, then at this digital level, you can affect behavior and so on. But for example, in certain diseases, GABA receptors or NMDA receptors or glutamate receptors are going to be selectively altered. And if we could simulate those effects, that could be quite powerful. So we've been starting a screen to look for molecules that have different properties that mimic natural receptors and ion channels. And here's an example of one that we've nicknamed Bizarro 15, which presents a linear IV curve when illuminated. When the light's off, it doesn't do anything. And it sets up a linear IV curve with a crossover potential around minus 50 millivolts when we illuminate with a defined wavelength of light here. And so this is something we're starting to work on. The currents are still small, so this is more of a proof of principle that we're working to rapidly improve. But it highlights the ability to look for molecules and to make molecules that can have these additional functions. And so to summarize the molecular part of the talk, one of the big open questions is how far can we go along these lines? And what are the kinds of things that can be enabled by selectively targeting the specific cell types or specific parts of cells? Now, the neurons of the brain occur, of course, in a densely packed three-dimensional environment. And this is there's not air in the brain. You have to deal with the densely connected mishmash or mess, as we call it in my lab, that we have to deal with in the brain. And so one of the things we want to be able to do is to activate neurons distributed throughout neural circuits. There are microcircuits both locally, but also there are likely to be lots of precise connections that transcend across many brain regions. And so one of the things we've been working on is to develop light devices that can allow independent optical control of dozens of points in the brain. Here's an example here, which is a hippocampal array that could target bilateral CA1, and every single fiber there is independently controlled. We can make them with fewer fibers too. This was picked partly for an art book. But one of the things that's being opened up by this is the ability to systematically screen through targets. There's sort of different ways to look at neuroscience. Top-down approaches where we want to begin with the behavior, and we might just want to go through the circuit and figure out when and how all the regions contribute. And bottom-up, where we might begin with a molecule or a cell type of interest and try to figure out how it contributes. One of the hopes is that we could use these kinds of tools to systematically go through the circuit, perturb them, let's say, in an unbiased fashion or in an exploratory fashion, and to use these tools in order to hunt down new circuits that are causally implicated. Of course, distributed recording is also important. And one of the themes that we're trying to pursue is whether we can develop structures that can allow us to pursue these kinds of distributed recordings as well in conjunction with these. And one of the things that we're encountering is also that in vivo work, you saw the beautiful motorized and robotic things that Alan showed in his talk. In vivo work is not a very automated or roboticized process yet. So we've started a program of research where we're trying to figure out how much of this can be made as high throughput and unbiased as possible. And so this started actually with an undergraduate, Stephanie Chan, who just started at Princeton in grad school, when she started building injector arrays to deliver viruses into three-dimensional patterns. So here's a triple injector that you can use to hit multiple points in the brain to deliver viruses to make them optically perturbable. And more recently, we've started working on ways of trying to automate the targeting of electrodes and other devices as well. So this is sort of a stamping device that you can dip in biocompatible ink and stamp out on the skull the locations of where you want to insert different things that it can be inserted in a high throughput fashion. One of the things that we're trying to work hard on is also to push on the delivery of light into the brain. So one of the things we're working on with Cliff Fonstad at MIT is the ability to take a probe that's about the size of an optical fiber or an electrode, and to be able to control all of the points along the axis of it, because that would allow us to minimize brain damage and ultimately to be able to perturb many sites in the brain. So you can see an actual photograph of one of these devices in action. What you see here are waveguides that descend into the brain, and then we've fabricated little mirrors at different points along these waveguides, so that each of them will terminate at a different depth along the brain and allow us to control all of the points along a given insertion track in the brain. And one might imagine that these might be very useful for trying to tackle problems where one wants to look at entire neural circuits. Obviously, this is just an illustration. The devices aren't that identical. But one can imagine making arrays of these, much in the same way that silicon probes and other strategies have emerged in neuroscience, and be able to go through the brain and perturb both individual sites and in combination different points of the brain to assess their causal functions, and potentially to also see what combinations might emerge as important. Recently, we've also started trying to figure out how to integrate electrodes into these 3D arrays. If we can fabricate them right down these probes, that might allow us to kind of kill two birds with one stone and be able to develop devices that have full integrated three-dimensional perturbation and recording. And so we've started to play around with fabricating our own silicon probe style electrodes that can be fabricated directly onto these as well. Finally, the last technology slide I want to talk about is also the idea of moving high throughput, not just larger within the brain, but also across brains. And so one of the recently graduated master's students, Christian Wentz, has been building fully wirelessly powered and controlled devices. Here's an example where one laptop can control 83 animals at once. And so we're interested in seeing whether this can be used to do high throughput assessment of the causal functions of cell types and circuits in animals. And potentially this could find different applications, not only in science, but also maybe in applications outside of science in the pharmaceutical industry or others. I want to close on one, so to summarize the hardware part, I mean, one of the big open questions is how systematic can perturbation be made? Perturbation is not usually thought of as a very parallel or high throughput thing to do because, when you perturb something, unfortunately, you can't really perturb lots of other things at the same time and still be completely unambiguous, as opposed to, let's say, recording, which might be able to scale up, very, very high. But there might be ways to work around this by thinking about neural circuits and about behavior, which would allow us to find ways to parallelize and scale up and make these things more high throughput. A final open question that I thought would be fun to think about is, if we have the ability to control these circuits, what are the practical considerations for making therapeutic inroads? And one of these stories I want to talk about was some work that's being led by Alan Horsager at the University of Southern California. And we're just collaborating with them, but he kindly is letting me show these slides, which is trying to figure out if you can have this kind of control, what kind of translational path might emerge from a neuroprosthetic standpoint. And the disease he's chosen to work on is retinitis pigmentosa, a form of blindness in which photoreceptors are lost. Here's a normal retina with all of its different histologically evidenced layers, including photoreceptors and lots of interneuron layers until you get to the final output layers that project to the brain. And here you can see the photoreceptors are gone in this patient's retina, but the rest of the neurons are still there, if a little bit in disarray. So one of the ideas is to take these photosensitive molecules and basically to convert the rest of the retina into a camera by photosensitizing a given layer. And in order for this to work, we collaborated with Bill Houseworth, who, along with Arun Srivastava, had evolved some new viruses that allow very, very high levels of infectivity in the retina using viruses such as AAV that are highly under consideration in the human gene therapy field. So as a disclosure, I'm a science advisor to a company that's seeking to commercialize this. But I thought I would show you some of the videos that Alan's taken here. So this is a blind mouse. We use different lines of mice, and it's trying to navigate in a maze. The maze has a bit of water to motivate the mouse to move. The goal is, in this case, is to get to a platform underneath the light, but you can imagine that the task can be more complicated by putting the platform in other places. And the mouse is solving this, because mice are smart, but in a brute force fashion and not using vision. And so, he's basically learned that, in order to find this platform, you just go into every one of these little holes one at a time, which is interesting for other reasons, but not the focus of today's lecture. And here's another mouse. I won't bore you with all this, but except to emphasize that we're looking at lots of different strains of mice in order to try and figure out what the principles are of translating these kinds of things. And then here's a mouse that was blind a few weeks before, but is now able to find targets. Now, exactly what the animal is consciously perceiving is not clear. That requires a lot of experimentation even to begin to get at. But what is clear is the animal is able to make use of visual information to some extent here. Another thing that we're trying to think about is how do you analyze the safety of these reagents and optimize the reagents for long-term health. So this is my last slide here. One of the things we've been doing is collaborating with labs such as Bob Desdemona and Grabio's labs, where they're working on non-human primates, and to look at cell expression of these molecules, since algal genes and archaeal genes and fungal genes usually aren't found in the brain. And what we found so far, both for the excitatory opsins, as shown here, and also for the inhibitory opsins, is that you can identify opsins that express well, that will last for months or even years without any evidence of cell death, immune cell infiltration, disarray of dendrites or axonal arbors, gliosis, all that good stuff that are the canonical markers of worry. And to the extent to which these early studies point towards potential safety in a translational context because of the non-human primate similarity to humans, potentially these are pointing the way towards therapeutic possibilities. So there's not really time to, in a short format talk, to go through every single person's contribution. I think I mentioned some of the key collaborators, but I'll leave this slide up and we can take questions, I guess. So thanks.